Chapter 5, Photosynthesis. The learning objectives for this topic are: define photosynthesis, compare and contrast the basic chemicals and structures involved in photosynthesis in prokaryotes and eukaryotes, describe the components and function of the two photosystems, PS1 and PS2, the PS stands for photosystem, contrast cyclic and non-cyclic photophosphorylation, contrast the light-dependent and the light-independent reactions of photosynthesis, and describe the reactions and products of the Calvin-Benson cycle. So in this topic we're going to talk about photosynthesis in general. We're going to talk about chemicals and structures that are required, that are found in photosynthesis. We're going to talk about the light-dependent reactions, cyclic photophosphorylation and non-cyclic photophosphorylation, and then we're going to talk about the light-independent reactions, or carbon fixation reactions as they are also known. So let's talk about photosynthesis in general. Photosynthesis is an anabolic process. Remember, we're taking smaller molecules, making bigger molecules out of them, that uses light as the energy for the process. So it can be divided into two sets of reactions. We have our light-dependent reactions, which directly use light energy, and then we use the light-independent reactions, which use the energy generated from the light-dependent reactions. So in the light-dependent reactions, we generate ATP and NADPH. Remember NADPH from the Enter-Dewar-Hoff pathway and the pentose phosphate pathway. Remember, this is an anabolic process, so we're going to be using NADPH. So you can think of the P for photosynthesis, or P for plant, because photosynthesis is the biggest and best of all anabolic processes. Now on to light-independent reactions. CO2 is fixed to larger molecules. So we take CO2 and put it onto a carbon skeleton. And the end product, carbon skeleton, is G3P. Remember G3P, our good friend from glycolysis? Yeah, GP3 is showing up all the time. Now that we know about the general process of photosynthesis, let's talk about chlorophyll and thylakoids. So chlorophyll is the molecule that makes photosynthesis possible, at least the light-dependent reactions. So we have a long hydrophobic tail that enables chlorophyll to be embedded in a membrane, and we have this active site which has a porphyrin ring with a magnesium in the center. Now the magnesium is important. So this is a molecule of chlorophyll A, which is found in plants, protozoa, algae, and it's also found in cyanobacteria. So actually, all of these eukaryotes got their chlorophyll A from forming an endosymbiotic relationship with cyanobacteria. So it makes sense that they would have chlorophyll A, and chlorophyll A absorbs in the 425 and 660 nanometer range. Now let's look at some bacterial chlorophylls. I suppose we could call chlorophyll A a bacterial chlorophyll, but we're not going to confuse the issue with that. So these are chlorophylls that are found in independently living bacteria. That would be green phototrophic bacteria and purple phototrophic bacteria. And bacterial chlorophylls absorb light in the 350 and 880 nanometer range. So they're not competing with the chlorophyll A containing bacteria. So the main difference between bacterial chlorophylls and the various forms of chlorophyll A are what spectrum of light they absorb. So let's go to structures. So I mentioned that because of this hydrophobic tail, chlorophyll can be embedded, whether it's bacterial chlorophyll or chlorophyll A, like in this diagram, they can be embedded in a membrane. And we have a tendency to get a whole bunch of them together into what we call a photosystem. Now we have two photosystems, photosystem 1 and photosystem 2, named in the order they were discovered, and they are found in the thylakoid membrane. Whether you're talking about independently living bacteria or whether you're talking about a chloroplast inside of a plant cell. This is what a thylakoid looks like in an independent bacterium. These membranous folds that are embedded with chlorophyll are infoldings or invaginations of the cell membrane. Cool, huh? So these are not separate structures in either bacteria or in chloroplasts. They are considered to be a part of the cytoplasmic membrane. Let's look at our first light-dependent reaction, which is cyclic photophosphorylation. Remember, this is phosphorylation, or putting a phosphate group onto, in this case, ADP, through the use of light energy. Light comes in, hits the reaction center, and eventually it hits an electron in the magnesium. So that causes the electron to become excited, and it hops up to a higher orbital. It can either be removed like it is in photosynthesis, or it can drop back down and release light back at a different wavelength, which is what happens when you shine UV light on it. So we're going to say that the electron is taken from the magnesium and gets passed to this iron-containing enzyme onto a cytochrome, which, now this is an electron transport chain, just like what we talked about in aerobic respiration. So the cytochromes here use the energy from the electron to take hydrogen ions and pump them to the other side of a membrane. Is this sounding familiar? It should. So this electron gets passed to a copper-containing enzyme complex, and it goes back to photosystem 1, goes back to home base on the magnesium. Another photon comes, energizes the electron, and it makes the circuit again. Meanwhile, the cytochromes are pumping hydrogen ions, or protons, to the other side of the membrane. In this case, it's the exterior of the prokaryote, or the thylakoid space of a chloroplast. Why are we generating this proton motive force? Why are we undergoing chemiosmosis again? Because we've got ATP synthase here. It's just like the ATP synthase that we find in aerobic respiration. Interesting, huh? As a review, the hydrogen ions come in through a hole in the ATP synthase. They're wanting to come to the area where there's fewer hydrogen ions, that they're wanting to undergo osmosis. The ATP synthase turns, ADP and inorganic phosphate are smooshed together to make ATP. So cyclic photophosphorylation is used when the organism needs ATP. Now, some bacteria only have photosystem 1. They do not undergo non-cyclic photophosphorylation, which we're going to talk about in the next slide. They can only generate ATP, but they use this ATP to cause their flagella to move. That brings them up and down the water column so that they can get close to the light, so it's still a benefit to them. So now let's talk about non-cyclic photophosphorylation. Just to give you a reference, here's photosystem 1, here's our iron-containing enzyme complex, which isn't used here. Here's our copper-containing one, here's our cytochromes. We start in non-cyclic photophosphorylation with photosystem 2, which was discovered and named second. So light comes in, a photon hits that magnesium, the electron is excited, moved to a higher orbital, and it gets moved down electron transport chain, the energy of which is used to pump hydrogens to the other side of the membrane. So far, so good. So this electron continues on to photosystem 1 and replaces the electrons that have been removed from the magnesium through the rest of this. Okay, so the magnesium gets another electron, it's been worn out through this electron transport chain, gets another boost of light, goes to this iron-containing enzyme complex, and then it ends up at NADPase. This is an enzyme that takes this electron, puts it on NAD+, causes it to be reduced to NADPH, which goes on to the Calvin-Benson cycle. NAD+, in non-cyclic photophosphorylation, is the terminal electron acceptor. Takes that worn out electron, removes it off to another process. Let's come back. It's lost an electron. It's going down here, it ends up on NADPH, and it's going off, and it's never going to see that electron again. Well, once the magnesium has lost an electron, it's going to hold on to the rest of its electrons even more because it's got a positive charge. So we have to replace this electron somehow. Well, what photosystem 2 does is it takes water, splits it, takes some electrons from the hydrogens. The individual oxygens pair up as atmospheric oxygen, and the process releases oxygen into the surrounding environment. Oxygen is a waste product of non-cyclic photophosphorylation. It is oxygenic. It generates oxygen. Isn't this cool? It's kind of backwards to how aerobic respiration goes. It uses water to produce oxygen, whereas further down the membrane, we may have an aerobic transport chain that's using the oxygen to generate more water, which is used by the photosystem too. Isn't that cool? Meanwhile, cytochromes are still pumping hydrogen protons, hydrogen ions, to the other side, and ATP synthase is still making ATP. So, from non-cyclic photophosphorylation, we get NADPH, ATP, and oxygen as products of the system. Now the question is, where does this NADPH and this ATP go to? The Calvin-Benson cycle. So here is the Calvin-Benson cycle. It's a cycle just like the Krebs cycle, but instead of breaking down molecules, it's making bigger molecules from smaller ones, namely CO2. We start out with ribulose biphosphate, or RUBP, and there's an enzyme called Rubisco that takes the CO2, puts it onto this 5-carbon molecule, and we get an unstable 6-carbon intermediate that breaks down into 2 3-carbon molecules. Now you'll notice that there's a 3 and a 6 going on here because we can't do this, just one. We have 3-phosphoglyceric acid. Do you remember this from glycolysis? Yep, it was there. So we use some of the ATP from the light-dependent reactions to change this into 1,3-biphosphoglyceric acid. Also, we saw this one in glycolysis. We take the NADPH to reduce the 1,3-biphosphoglyceric acid, and eventually you get G3P, our good friend G3P. So we pull some of this out of the cycle, put 2 of them together to get glucose 6-phosphate, remove the phosphate group, and you have glucose, which can be used as an energy source when the light's not shining. We can use it to build other carbohydrates. We can use it to do all sorts of good stuff. Meanwhile, we've still got to have some GP3 to go through the process of regeneration into RUBP. So it takes more ATP. Then we take some more CO2 and we go through the cycle again, producing more glucose from the G3P. They used to call these reactions the dark reactions because light is not directly involved. Well, the Calvin-Benson cycle can't run in the dark because we don't have the supply of ATP and NADPH that we're getting from the light-dependent reactions. It's kind of like you're shutting off the electricity to the plant. The factory has got to have electricity. So then they named it the light-independent reactions. Well, they're not really light-independent because they're dependent upon the light from the light-dependent reactions. So personally, I prefer carbon fixation reactions, but this textbook uses the term light-independent reactions. So you can go with that. So that's it for this topic. Here are some reminders of what I wanted you to learn from the topic. And next topic, we're going to talk about other anabolic processes besides photosynthesis.